Hi everyone, welcome to MBIOS Research Insider 5. My name is Yita and I will be your host for today. We are also broadcasting our session live on MBIOS Facebook, so please feel free to share the live stream with your friends and family. We will have a dedicated Q&A session towards the end of the presentation, so should you have any questions for our speaker, please feel free to type out in the chat box. Our ability to host events for our student members was made possible by the generous support of our sponsor, UK Education Centre. UK Education Centre is the leading education agency in the UK who provides free support and services for students who are looking into higher education in the UK, such as university applications and also visa services. If our student members are considering to apply to UK universities, we would recommend you to get in touch with UKC for a free one-to-one consultation today. I would also like to take this opportunity to introduce you to MBIOS Certificate Scheme, which allows current MBIOS members to receive a certificate for their participation in any of our events. There are currently 3 certificate tiers that can be claimed, and these tiers work as an accumulation. You will have to participate in the respective numbers of events as per the tier outline to claim your certificate. We would also need a proof of your attendance in the form of a screenshot for you to be able to claim the certificate. If you're interested to be part of the scheme, kindly check out MBIOS social media for more information. Without further ado, please allow me to introduce our speaker for today's session, Pei Cing. So Pei Cing graduated from the University of Sheffield back in June 2021 with a Bachelor of Science in Biochemistry and Microbiology at the University of Sheffield. And during her time in Sheffield, she was a recipient of the International Merit Undergraduate Scholarship and also the Faculty Undergraduate Scholarship. Pei Cing graduated top of her class and she was subsequently awarded the NLD BBSRC-funded Doctoral Training Studentship to pursue her PhD project at the University of Liverpool. Her PhD project investigates bacterial microcompartments through the lens of structural biology, bioengineering, and biochemistry. Now, I would like to invite Pei Cing to take the floor and provide us with further insights into her academic background and also her PhD project. Let's welcome Pei Cing.
So hi everyone, good afternoon from Liverpool. Thank you for joining. Thank you MBIOS for inviting me to speak on today's Research Insider session. So yeah, I am Pei Cing. People here call me PC because they can't pronounce it correctly. I am a PhD student at the University of Liverpool. I'm currently halfway through my second year. And as Yita has alluded to, I do a lot of, I would call myself a molecular biologist. To summarize my PhD in 4 emojis, I take DNA from bacteria, and then I use E. coli to grow them up, and then I investigate my proteins under the electron microscope. I would say currently I'm still more of a protein biochemist because structural biology can be quite technical and also can get quite expensive as well. But I have started doing a bit of structural biology, and hopefully I will be more of a structural biologist by the time I graduate. My PhD is fully funded by the NLD BBSRC DTP. And I guess today I'll be telling you, I'll tell you a bit about my background, how I got to where I am today, just to give you a bit of context. And I guess I'll focus more on what are BBSRC studentships, because I guess a lot of you must be interested in applying to one. Tell you what they are, how to apply, especially as an international student, and why it's really great to be on one. And then I'll touch a little bit about my research, my PhD research on bacterial microcompartments. I won't be showing any data from my PhD today because most of them are unpublished, but I will be telling you about the methods that I use to study them and why we want to study them in the first place. So this is a structure, a really nice structure of a type of bacterial microcompartment. And you can see that it looks very much like a virus, and it's entirely protein. So they're really cool things to study. But just to give you a bit of background, I did my SPM in high school, as I imagine many of you must have done as well. And it was towards the end of high school, like in Form 4, Form 5, that I started to study, that I decided to study seriously for SPM. And it was then that I decided that because the lessons at school were quite dry, so I decided to try a different approach to study science, and I went on YouTube and found all these really nice educators making really interesting videos about science. And I won't go into them today. I'm sure some of you might have seen them as well, but they are the reason why I am doing science, why I am doing a PhD in the first place. And they really gave me the inspiration to want to study, to want to discover the things that they were talking about in those videos. And then in high school, I was also really active. I was part of the Chinese orchestra in high school. We often had annual concerts and also went for national competitions. So that gave me a lot of extracurricular activities, extracurricular points to get me a place in the Malaysian matriculation program. So you might be thinking, why is she going for matriculation when she wants to study science? Because, you know, the government sends out people overseas to study science. I am overseas doing science now. But I guess I did A-levels. I did a tester of A-levels before the matriculation list went live, before I knew that I was admitted onto the matriculation program. And I decided that I didn't want to be paying that much money to study locally. And I also thought that if I had to study science in Malaysia, I wanted to do it in a local uni because I felt like I would have access to more facilities, and I would be exposed to the local network, to the local scientific network more if I did it at a local uni. So I went to matriculation and I had an absolutely great time. So I learned about different cultures when I was in Malaysian matriculation. I still got to play my instrument, and I really enjoyed having that community feel in matriculation. So this is kuliah. This is the place where we have our lectures. It's an absolutely big hall. You can have up to 5 classes of students here. And then because in matriculation you stay in dorms, and often those dorms are in places that are quite far from town. And so you are surrounded by a lot of beautiful nature. So this is the view from my dorm. I would walk to lectures every day here. And you have absolutely beautiful views of sunset or sunrise in matriculation. So that I had a really nice experience. I'm glad I went there. But one thing that I think I managed to experience firsthand is how I guess I was a bit disappointed by how people weren't really interested in studying science in general. So most of the people were doing science just to get into, well, not just to get into medicine, or for example, engineering, which is completely fine. And if that's what you're interested in, that's great. But I wanted to be surrounded by an environment where people were interested in understanding the fundamentals of science. So I then decided that I would actually give it a shot at applying for overseas universities. And in particular, I applied for UK universities through UCAS, through the standard procedure. And I searched for universities that would admit matriculation students, and there are quite a few around that time when I applied. And I wrote a very earnest, I think it was a personal statement at that time, explaining why I wanted to study science. And I got into Sheffield, which was what Sheffield was my first choice because I was attracted to how flexible their molecular biology degrees were. So I enrolled into a biochemistry program, but I graduated at the end with a biochemistry and microbiology degree. And so because as I'm sure many of you know as well, if you study in the UK, the tuition fees and the everyday expenses can be quite expensive. So I applied for scholarships. When I applied on UCAS, I think I had to submit an essay as well to Sheffield, explaining why I wanted to be considered for the International Merit Undergrad Scholarship and why I wanted to study at Sheffield. And once you get this scholarship, so it was a 50% tuition, a reduction in your tuition fees. So once you get that, you just have to maintain a 2-1, so that's a 60% average. And then you would just be awarded the scholarship for the rest of your degree. And the faculty undergrad scholarship, I'm not sure if these have changed since then, but it was awarded automatically to internationals as long as you maintain a 2-1 as well. So I would say around that time, Sheffield was quite generous with scholarships. with international scholarships compared to other universities. So I did my degree in the Department of MBB, Molecular Biology and Biotechnology. This is FERVCO. This is where most of the research labs are. So imagine how nice it must be to be sitting in one of these labs and just looking out, doing a research on a really sunny day. And one fun thing that I wanted to show you is the para labs. So that's where I did my undergrad labs in. And this is not a coincidence. I think back then Sheffield has sort of collaboration with Malaysia. It was not really explained to me why, but they named the undergrad teaching labs the para labs. So that was really cool. Molecular biology includes things like genetics, microbiology, also cellular biology as well, and biochemistry. And in your first year and the first half of your second year of your degree, you sort of have to study everything to give yourself a good enough background to then go and specialize in something. So I found myself gravitating towards biochemistry, and in particular biochemistry and structural biology. So that's the study of proteins. So you would be looking at protein structure and how that relates to function, how different proteins interact. And in particular, biochemistry is very useful for things like drug discovery and development, where you would, if you have a structure of a receptor that is implicated in disease, you can then design a really good drug to inhibit that receptor, for example. So I got a summer studentship to do a 3-month placement in a research lab in Canada. I would be doing structural biology on a membrane protein, which is part of a membrane protein in bacteria, which is implicated in pathogenic bacteria. And unfortunately, that summer was the summer when COVID happened, so it got cancelled. And I guess my personal tutor took pity on me, and we applied for another funding from the Genetics Society. So for people who don't know what personal tutors are, I'm not sure if other universities have them, but they are this academic person in the department that is given to every undergraduate. And then you can, basically you have to write essays for them to mark, but you can also discuss personal or academic issues with them. So my personal tutor was really great. He decided to do a project with me that summer, and we were investigating pigment biosynthesis in bacteria that photosynthesize. And what that entails is, what molecular genetics entails is that we were making mutants, we were knocking out genes that we thought were working in this biosynthetic pathway, and then we were seeing how these mutants are affected in their ability to produce these pigments. So this is cyanobacteria. It produces green pigments, whereas this is Rhodobacter, it produces red pigments. So, although this wasn't biochemistry, this wasn't structural biology, this gave me a really good experience in terms of cloning, designing primers, and also genetic manipulation. So those are very good skills to have in any aspect. If you're trying to do a cellular biology or molecular biology work, you will be doing cloning quite regularly. So that gave me a really valuable experience. And around that time, in the summer between my second and third year, I started researching about PhD programs across the world. It wasn't just the UK. I also looked at universities in the US and also Europe. So that's one thing. If you really want to do a PhD, it's important not to limit yourself to one area, one country. Japan also has a really good international PhD program. And then I started thinking about which area I wanted to specialize in for my PhD. And then so I decided that I found microbes more interesting than humans. So this is entirely subjective, but microbes, they are more metabolically diverse than humans. And I guess as COVID has shown us, it is important to understand the microbes that are dangerous to us as well, so that you can treat them. I started applying to PhD programs in this area at the beginning of my third year, and then I changed my degree to biochemistry and microbiology as well. That it didn't have any effect on my visa whatsoever. It was quite an easy process. And then for my third year project, I guess it was also because I did a summer project in bacterial photosynthesis, but I got increasingly interested in this area. You know, cyanobacteria, for example, is the precursor of the chloroplast, and the red bacterium, the Rhodobacter that I showed you earlier, it is the precursor of mitochondria in living cells. So it gave rise to life on Earth, and most of the oxygen that we breathe in are also produced from bacterial photosynthesis. So just to give you a really brief overview of my third year project, I'm sure you learned this to some extent in your degree. So you have the photosynthetic electron transfer flow produces energy for the production of food. And in particular, we were interested in two proteins in that photosynthetic electron transfer pathway in cyanobacteria. So we were doing bacterial culture. This is how we actually grew them in the lab. We were making mutants of them, and then we were purifying these proteins. So this is actually, I'm showing here an assay for this little plastocyanin protein. So plastocyanin has a copper cofactor, and when you oxidize it, it turns blue. So that's how you know that you have your protein there in the sample. And one really cool thing that we did to characterize the interaction between those two proteins is by peak force quantitative atomic force microscopy, or AFM for short. So I think AFM it's more commonly used as a as a physics method, and people use it to characterize materials. People use it in physical chemistry, material science. This lab has adapted the method for studying protein-protein interactions. And the way that AFM works is that you have this cantilever, you have a tip attached to a cantilever, and then you put your material on the surface, and you sort of drag the tip across the surface, and it bounces across the material, and that will give you an indication of how rough the surface is, how thick they are, how hard they are, the elasticity of the material. In this lab, they attached plastocyanin to the tip of the cantilever, and then this big membrane protein, which is its interacting partner on the surface. And by bouncing the tip across the surface, the resistance by which the cantilever experiences when you try and take it off the surface is an indication of how strong the force, the interaction is between those two proteins. So this is a picture of the surface of the substrate, that these are all the different proteins sitting on them. You can tell how big the proteins are from looking at this, and then they also give you the force of the interaction in piconewtons. And I'm just showing this just to tell you how much my interests were influenced by the research in our department. So I thought I was going to go down that route of pure biochemistry and structural biology, but got interested in bacterial photosynthesis. So this is something that you might want to think about if you're already in your undergrad and you're thinking of doing a master's. Maybe you want to think about which university has the labs that will help you get to which area you want to be in your academic career, because you will have access to the facilities that are available at that particular university. So it is very much influenced by what sort of research is going on in the school. And so here I am. This is where I am today. I started my PhD after graduating from my bachelor's. So I'm at the University of Liverpool. I do a lot of protein purification. I grow them up in E. coli, I purify them. I'll talk about them a bit more later on. And then, I guess, as I said, I use electron microscopy to look at my proteins. This is a picture of the sample grid under a standard transmission electron microscope. And this is a vitrobot, which is what you use when you want to prepare the sample for more high resolution imaging. As Iza has said before, and as I've alluded to in my introduction, I am on the NLD BBSRC doctoral training partnership. So I'll now tell you a bit about what a BBSRC DTP is, how do you apply to one, and what sort of perks that you get of being a DTP student. But just want to say a disclaimer, these views are completely my own. I am not representing the BBSRC in any way. I am purely speaking from experience, and if you want like factual information on these programs, you can really easily find pages online explaining to you the fine print. So, just in case I don't want to get sued. Yeah, so the BBSRC stands for the Biotechnology and Biological Sciences Research Council. It is one of the 7 research councils in the UK, making up the UK Research and Innovation. We just call it UKRI. And what the research councils do is that they take taxpayer money and they decide what research to fund, and they do it by way of grants. So that's what PIs in labs apply for, to fund their lab, to fund the running of the lab. But research councils also provide funding for PhD studentships. And they do it by providing money in the form of doctoral training partnerships. Doctoral training partnerships are essentially training programs for PhD students that are funded by UK research councils. They provide you with a stipend. This is probably the one that you're most interested in. Currently, it's £16,000 per year, which is about £1,400 a month. It will be a bit higher if you're based in London, because London is expensive. They will also provide the university with tuition fees, but so this goes straight to the university. You never see this. And you also get a research training support grant. So that includes things like conference travel, conference attendance, training, bench fees, all that science money to support your research, basically. Another type of DTP program is called the IK studentship, and it makes up a smaller amount of the overall DTP program. But what it does is that you do your PhD with an industrial partner. So you would do part of your research in an academic lab, and you would also do it with the industrial partner. So, for example, if your IK studentship is with GSK, your PhD project would most likely be less fundamental and more applied. So you would be probably studying a protein that is of interest to the pharmaceutical company. And then, so with the BBSRC DTP, well, not just the BBSRC, with any DTP, there is no bond after you graduate. But you do have to progress and finish your PhD. In the UK, this is usually 4 years. Although if you did your PhD during COVID, you would get an exception. You would be able to extend your PhD. But normally you would aim to finish within 4 years. And in 2020 was the year when I first started applying for PhDs. And that was when they opened up the call for international students. So in the past, so before 2020, the DTP studentships were limited to European applicants. So that included UK students or European students. But after Brexit, they opened up the application for everyone, which is good and bad because then the competition increases. But I will tell you a bit about how you can make yourself competitive for these studentships later. These research councils, they gave a pool of money, a sum of money to partnerships between institutions. So that includes universities or research centers that are usually based in the same area. So for example, my DTP is coordinated by Newcastle, Durham, and Liverpool. So we are all up in the north. In Scotland, you have East Bio. And then down in London, you have Lido. In Oxford, they have their own DTP. And in Norwich, you might know of this if you're a plant person. They have their Norwich Research Park DTP. So each program is coordinated independently of one another. They have their own management board. And I guess all of them have more of a biological focus. The NRP DTP focuses more on plant science, but they all have their own training activities, their own application process, and they also determine the amount of studentships that they want to give out every year. So I thought it would be helpful to give you a timeline of the whole application process of the DTP studentship. So between October and December, that is when you start seeing PhD projects for the DTP program being advertised. If you're interested in a particular lab, if you're interested in a certain PI, you can most definitely stalk their Twitter to find out whether or not they have any PhD positions available. You can also find them on their websites, on your own websites, or you can just go to the DTP websites or the university websites to see a list of the available projects. Find a PhD is also really great if you just want to search for PhDs in a certain area or a certain or using a certain technique. And most often these DTP PhDs will be advertised as competition funded. What that means is that the list of projects that are being advertised will be more than the amount of projects that will be funded in the end. So you will be competing against people that are nominated for another project to get the funding from the management board. So that's what that means by competition funded. You don't get the money. It's not that straightforward. But yes, you will start applying between October and December. And because you have so many different projects that are available, I would recommend making a spreadsheet to keep track of everything. So in terms of documents, I would say it's pretty standard for any. It's the same as how you would apply to a normal PhD. You need to write a cover letter and a personal statement that's normally 1 to 2 pages long. You will need 2 recommendations usually. Sometimes they ask for 3. And if you're not from a UK or not from an English-speaking country, or not from a country that uses English as their primary language, I think they will ask for proof of English proficiency. But this is definitely something that you can double check from the website. So you can have a chat with the project supervisor after applying. If it's before applying, it's for you to have an opportunity to ask questions about the project. And also a good way for you to show that you're interested in the project. If you go and read the papers, maybe you can come up with ideas on how you could contribute to that project. But absolutely, it's a good way for you. It's also a good way for you to see if you would get along well with the project supervisor, because that's very important. If it's after you put in an application and the project supervisor emails you to say that they want to have a chat with you, usually that's the project supervisor. They might be thinking of putting you forward for the board interview. So they'll normally tell you that as well, that they'll put you forward. And they might offer to have a mock interview with you and also clarify any questions that you might have about the project. And then, so yes, the supervisor will have to put forward. So from this pool of people applying for that particular PhD project, the supervisor has to choose 1 or 2 people to then go forward for the board interview. So that's like people who are in charge of the DTP program for each university. So that's a formal interview, and you have to do, in my case, I had to do a short presentation. It was 3 minutes long. So you have to really train yourself to be able to explain your research really concisely. A short presentation of a research project that you did. And then you have Q&A session where they will ask you questions about your research project that you presented about. They might ask you to clarify certain methods, see whether or not you actually understand what you're doing. And then they will also ask you really generic questions. You can usually find these online, but they'll ask you things like, why do you want to do a PhD? Why are you interested in this particular DTP program? What can you bring to this project? Why are you interested in this university? Things like that you can find these questions online. And then usually it is around March or April that you hear back whether or not you've been offered a studentship. If so, you celebrate. If not, absolutely just keep applying. Some other studentships might have a second call around March or April, or you have like completely different PhD. You have completely different. You have another DTP program that only starts advertising in this period. So just keep on searching. And then if you get if you get a studentship, you would start your PhD in October, which is usually the academic year for a PhD student would start in October in Liverpool. I wanted to clarify this point here. So back when I applied, some DTP programs actually they will say on their website that they expect international students that are accepted to cover the difference between home and international fees, which is, I find a bit unfair. But sometimes if the supervisor is really interested in you and if they put you forward, usually they will try and find a way to help you cover the difference. And this is something that you can discuss with them as well. In my case, the DTP program just classed me for just university purposes, just classed me as a home student in terms of like fee paying status. I still had to apply for a visa, but it was just easier for them to say on my profile that I was charged home fees. And so I guess I just wanted to emphasize again, each DTP program has their own application process and the number of studentships that they offer. So you don't have to limit yourself to the BBSRC. If you're interested in the medical side of biology, you can look for studentships offered by the Medical Research Council, the MRC. These studentships are very popular. I find that they are more competitive than the BBSRC ones. But if you're more of a nature person, you can also you can look for NERC studentships as well. If you don't have to limit yourself to the UKRI, other international PhD programs that are available are offered by the MRC, LMB in London, I think. London or Cambridge, can't be sure. But this is absolutely top-notch. This is state-of-the-art lab. They have a lot of facilities there to produce really great research. They're more on the human side of things, more medical, as well as the Francis Crick Institute. And the Wellcome Trust also provides PhD funding for international students. And these run in the same manner as the DTP studentships. So they offer money to, I think it's group of universities rather. I think they offer money to a certain university rather than a group of universities. And then you also join a cohort of people in that same year. I got onto one, I was offered a Wellcome Trust studentship in Dundee. And I found that the stipend that they offered were more generous than the BBSRC studentships. So it was around £19,000 per year. Compared to the £16,000 that were offered by BBSRC. So that's just something to take into consideration if that's something that's important to you. But you might have sorry, lost my train of thought there for a bit. But yeah, DTP studentships are very competitive by itself, and it's even more so for international students, unfortunately. In my cohort, there were 20-something of students admitted onto the program, and there were 3 of us that were international students. So around 2 to 3% 2 to 3% spot for internationals. So how do you make yourself competitive? I would say as an international student, unfortunately, you have to destroy the competition. It will say on the website that they need a minimum of 2-1 in terms of your results. That is such a lie. You need to show that you are top of the year, or you have a strong track record, not necessarily top of the year. By that, strong track record is important because it shows that you have sort of the grit to progress through your PhD, and also implies that you have the background to be able to do research. In my case, I was awarded prizes from Sheffield in my 2nd and 3rd year, showing that I was top top of the cohort. So that's, absolutely something that you can see on your CV as well. And because the DTP is essentially going to invest in you, they're going to give you money to do research. They're going to give you a lot of money. So you want to show evidence that other people also have funded you as well. So in my case, that includes scholarships from Sheffield, but also if you've won summer research funding, definitely say that as well. Also the MyTex one, the Canada one that I was supposed to go to, that was really good. That was actually really good on my CV because it was an international scholarship. So, show that you've won different scholarships if you have them. Other things that you can do is to diversify yourself in terms of the techniques that you know and also the research areas. I think people would like it if you apply to a project that you already sort of have the background for, but you are absolutely not expected to be an expert in that particular technique. That's what a PhD is for. So it is always good to try out different things in your undergrad or your master's, learn different techniques, and that will really help you in your PhD, because I don't think you don't just use one type of method in your PhD, at least not in my experience. And then another thing that you want to do when you're actually applying is to be intentional and proactive. So what that means is that you want to think about the research area that you want to do your PhD in, which lab will it help you for what you want to do in the future after your PhD. And a really good way is to just, email the project supervisor and then show them that you're interested. If you do get an offer to interview, absolutely do mock interviews with anyone who is willing to do them with you. A really good, sorry, the best people to do mock interviews with are PhD students in the lab, because they've just gone through the interview and they will know what sort of questions they'll ask. And they're more available than lecturers, for example. Absolutely research the DTP program and the host uni as well. Know why you want to apply for them and the facilities that are available there. And then one last piece of advice that I would say is just to keep on applying. Don't just apply to anything. Apply to PhD programs that are suitable for you. But I did have to apply to quite a few. I applied to 9. I was about to apply to 10 because I definitely got the rejections first before the acceptances. I put 1 here because I got to the interview for 1 and I was rejected. But I took the experience from that and reflected on how I could have done better. And then I got 2 acceptances afterwards. So I just want to quickly touch on as well on how to decide on a lab, if that's something that you would like more input on. So absolutely the number 1 priority is to find a lab that will support you, find a PI that will support you, find PI where the mentoring style matches yours. So that means that you have to think about whether or not the PI is a hands-on or hands-off person, and which which one do you prefer? Do the lab members help each other? Do they support each other, or are they competitive? And if you have the opportunity to as well talk to past lab members, because see where they are in their careers, because that is often a good indication of how well the PI is to their students. Then you can start to consider whether or not the research and methods, the techniques that they use, align with what you are interested in, because often research interests can change over time, and it's not a bad thing that it changes. It's not a bad thing if it changes. If you want to be really successful in your research career, it might help that the PI is a absolutely giant in the field. But often I find that junior PIs help the PhD students more. And then, university rankings matter less at this point. You definitely want to go to a university that has the funding to support the research that you're doing, but it doesn't matter that much on the grand scheme of things. The most important thing is you need to find a lab in the city that you will be happy in, that you will be mentally stable in. So after scaring you off from the application process, I want to quickly talk about the joys of being, I wouldn't say the joys, but what the perks of being on the DTP program. So you have an induction at the beginning. So essentially you are joining a cohort of people that are also funded on the program. So you have an induction in the beginning where you meet the rest of your cohort, and then you get to share the research, share an introduction of your research. And then you have events throughout the years. Usually that's in years 1 and 2 of your PhD, because that's when you have the most time. The one that I really enjoyed last year was the DTP conference that they organize every summer. So I got to present my research there, and we got to have dinner at Durham Castle, which was where Harry Potter was filmed. So these events, you get reimbursed for travel, and then you get to stay in hotels and get to have fancy dinners. So that's just really something that's really nice and provides a good distraction from the stress of your PhD. Another thing that you get as well is you get to join data science and computational biology courses. They will provide you the courses. It's not like you can't just choose a course and then tell them to fund it for you. But these are really good courses, and they will cover the expenses for you. They also have, they cater to different skill levels. So they have a beginner and intermediate levels as well. And then another thing that I enjoyed was they do fund, if you need to, they do fund external visits to labs. So I went to Newcastle to learn how to analyze my data. And I also learned a really cool method to produce proteins outside of the cell. Another thing that is really great about the DTP program is this professional internships for PhD students. So that's basically, it is a requirement as part of being a DTP student, in that you have to do a 3-month placement. That is not, you have to do a 3-month placement at somewhere that is not related to your PhD whatsoever. So that can be in an industrial setting, or if you don't want to do science at all, you can do it in marketing, you can do it in policy making, or even media outreach. So anything that you would like to try, because not everyone gets to become a lecturer at the end. In my case, I am going to go on to do my 3-month placement at Signature, which is a drug discovery company in Nottingham. And I find that really nice. It's like comes round circle, because I thought I would be doing drug discovery. I thought I would be doing biochemistry, structural biology, in like drug discovery. And now I get to do it. I didn't get to do it as part of my PhD, but I get to do it in an industrial setting, which is actually even better, because I get to experience how drug discovery is from the pharmaceutical side. All right. So I've reached the end of that. And now I just want to briefly go on to talk about my PhD research. So I research bacterial microcompartments, or BMCs for short. They are these like structures that look like viruses. They are found in bacteria. So you might be asking, like, organelles. I thought bacteria didn't have organelles, but so this is actually quite a relatively new discovery in terms, like, compared to other, like, the rest of biology. We now know that they are widespread throughout the bacterial kingdom, and different bacterial microcompartments in different bacteria do different functions. So for example, in Salmonella, bacterial microcompartments are implicated in pathogenesis, whereas in cyanobacteria, which photosynthesizers, they help in photosynthesis. So these organelles in bacteria, they are different from, Golgi apparatus and the plasma reticulum in eukaryotes in the sense that they are entirely protein-based. So you have a protein shell encapsulating cargo within. And it is an entirely self-assembly process. So we think that there are two self-assembly pathways. The first one is that the cargo assembles before the shell, and another one is that the cargo assembles with the shell. Structural biology is actually one of the best ways to characterize these bacterial microcompartments. And Weng Ming actually gave a great talk on structural biology on a MBIOS Research Insider session. So I won't go into detail, but from structural biology, we now know that the facet of the shell, which is what's highlighted in blue here, is made from these hexamer and trimer proteins, and that the vertices of the shell, which are in purple here, are kept by these pentamer proteins. And these shell proteins have different pores in the center, and they have different sizes. The pore sizes are different, and the characteristics of the pores are different. So they confer different semi-permeability characteristics to the entire BMC shell. And then we know that the cargo is linked to the shell by these encapsulation peptides that are highlighted in gray. So bioinformatics has shown us that there are 68 different BMC types across 80% of bacteria. So you have different shell proteins that you can mix and match. Another thing that I haven't told you about is that these BMC proteins, they display structural plasticity and modularity. So what that means is that you can take a BMC shell protein from one bacteria and then mix them with other shell proteins from other bacteria, and then they'll form all these different shells. And then you can just, you can tailor them to have the surface charge that you want, or even, display antibodies or display receptor proteins, or encapsulate the drugs that you want for drug delivery. So this is actually really cool, and a lot of people are doing BMC bioengineering with these BMC shells. As well, we also know that these shell proteins not only assemble into icosahedral structures, they also form tubes and also sheets. So you have different molecular scaffolds that you can use to tailor to your own biotechnology purposes. So it is a really cool field. And the way that I study them, so I do a lot of protein purification of these BMC shells. And so I take the genes from the bacteria that are producing that produces these shell proteins. I clone them into a plasmid that E. coli can use to produce these BMC proteins on a larger scale. I then harvest them and I lyse them using ultrasonic waves. By lysis, I meant that I break apart the cell membrane and release the proteins inside. I then further purify the proteins using sucrose ultracentrifugation methods, and that's probably something that you have heard about before as well. So I first pellet my proteins using a sucrose cushion, and so I spin the lysed cell light. After lysing my cells, I spin them at really high speeds on the sucrose cushion, and that lets me pellet my BMC shell proteins and also other proteins as well that is dense enough to pass through the cushion. And the cushion is sort of there to soften the blow of pelleting process because we are spinning them at very high speeds. I then take the pellet and I resuspend them, and then I further purify them with a sucrose gradient, again spinning at really high speeds, and then different proteins will fractionate out based on their density. And then I take the fraction that is enriched for my protein, and then I further purify it on an ACTA machine. So this is what people call fast protein liquid chromatography, and it lets you separate proteins on the basis of charge, for example, or size, or well, the best method to actually purify proteins is by having tags on them. So these affinity tags on your proteins will be recognized by the column, and that will let you separate out your protein from other contaminating proteins really easily. And then after I purify my protein, I then characterize them. I then quickly run an SDS page gel to look at the size of my protein, how pure the protein is, and also whether or not I've even got my protein. So this is a really quick way to just look at your sample. And then if I've got my protein, then I go on to transmission electron microscopy. I stain my sample with a heavy metal stain. So that's negative staining. It stains the outside of the protein. And then we shoot electrons at them. It produces this really nice image. It is a low resolution image, but it will give you information on the sizes of your proteins and how pure your sample is. Because if you've got other contaminants in there, it will show up on the image. And then this is also really quick. You can do it in a matter of hours. And then if it's really good enough, then we go on and do the expensive, we go and put it on the expensive equipment. So that's cryogenic transmission electron microscopy. So you freeze your protein really quickly in a process called vitrification. So this is done on a vitrobot. And that quick freezing process prepares your sample for high resolution imaging, but it also maintains your protein in its native state, so it's not damaged. And then that will let you, if your sample is good enough, that will let you look at the protein-protein interactions within the whole bacterial microcompartment, look at its structure, and as well as its size. So that's high resolution imaging. This is a grid of an example of a cryo-EM grid. And this is what we have to screen through these grids to find the best spot on the grid to image and get data for. So that's very high cost, very, very high cost, but we do all these optimization steps before before deciding to do this. So we prepare a good sample for it, and it will be quite quick as well if you've got a good sample. And that will, you'll probably be able to have a good structure within a matter of weeks. So that's, I think, yeah, that's all that I have for today. Thank you all for listening.